So we must be live now. Excellent.
Excellent.
Hello, everybody, and welcome to the second Sussex Vision series talk within the Worldwide Neuro Initiative. I'm George Kafetsis, currently a master's student at the group of Thomas Euler, and soon to be PhD student with Tom Baddell. As your host for today, I would like to first and foremost thank the organizers and more specifically Tim Vogels for the logistics of this talk, but also, and probably even more importantly, for this initiative towards a greener and more accessible seminar world. The extraordinary, enduring situation we all witness provides an excellent opportunity for discussions and for much needed and science-led changes. Getting back, however, to the reason we all gathered here today, please allow me to introduce our guest, Dr. Gautam Awatramani from University of Victoria in Canada. Upon acquiring his PhD in physiology and biophysics from State University, New York, he worked as a fellow in the labs of Lawrence Trussell, Timothy Murphy, and Botond Roska, during which time he was investigating neural activity and transmission down to synaptic resolution. After a couple of years at Dalhousie University, he moved in 2012 to University of Victoria, where he is located ever since and currently holds the title of Associate Professor. At his lab, they use optical and electrical recordings together with pharmacological or optogenetic manipulations in order to investigate neural circuits under physiological or neurodegenerative conditions. And today we will be hearing about their latest and I'm pretty sure exciting findings. Without any further ado, please all welcome Dr. Awatramani.
Good morning, everybody, or good afternoon or good evening. So can you see my screen? Or do I have to do a share screen still?
Share screen first.
Share screen first, right? Okay. There we go. Can you see the screen?
Great, yeah.
Okay, hi. So I'm a little bit nervous. It's the first online talk, and I live on a beautiful island where the internet connection is sometimes spotty. So I apologize in advance if I have some connection issues. I'm also nervous because of late my 5-year-old daughter has been especially fond of me and might come barging into the room. Poor thing is socially isolated and only has a dad to play with. But hopefully those aren't issues today. What I'd like to talk about today is work in our lab that we've carried out in the last few years regarding the role of inhibition and excitation in the direction-selective circuit. So typically, can you see the pointer?
Yes. Even better.
Yeah, so typically you have, in a classic model of a neuron, you have inhibition and excitation that are collected by the dendrites that funnel down to the soma, and then it's the net sum of inhibition or excitation that either cause the cell to fire or not, okay, after reaching a certain threshold. So it's the collective sum of inhibition and excitation at the soma. More recently, it's been recognized that some important steps of integration might happen at the dendritic sites themselves, at distal dendrites, and accessing these is quite a challenge. So today I'm going to tell you our work in which we make somatic and dendritic recordings, and we see how we can piece those together. So in the first part of my talk, I'll just give you a basic introduction of the direction-selective circuit and what the basic mechanisms are, emphasizing the role of ACH. Okay, this work has been done a lot by Laura and Santosh. In the second part of the talk, I'll talk about the spatial temporal dynamics of ACH at single synapses, at single cholinergic connections between the starburst and the DS ganglion cell, or the direction-selective ganglion cell. This is going a little bit into the weeds, and you'll see we go quite into detail, but then we'll kind of zoom out and try and understand how these spatial temporal dynamics relate to directional computations. Okay, and this work has been done by Akihiro Matsumoto, a postdoc in Keisuke Yonohara's lab in Denmark, and a favorite postdoc or graduate student, I should say, David Burson, who did a lot of the SP serial block EM, and then a lot of dendritic imaging by Varsha Jain and Ben Murphy, and some modeling by Jeff Rosenroll. And the basic conclusions I want to convince you about are that the steps of integration happen not at the whole cell and not even at single dendrites, but little pieces of dendrites, 5 to 10 microns in length. Okay, and these are the origins of the nonlinearities that create what we'll show you to be dendritic spikes. I'd also like to convince you that both ACH and GABA are tuned for direction at the level of single dendrites. So it's not just the null direction GABA that's tuned, but ACH and GABA are tuned for direction. And finally, we'd like to show you that the computations that happen come from a variety of mechanisms that are distributed heterogeneously along the dendritic tree of ganglion cells. Okay, so let's give you a little brief introduction. So all this started a while back when, almost 50 years ago, when Barlow and Levick were characterizing the receptor fields of ganglion cells. So when they used small spots and flashed them over the receptor field, what they noticed were on and off responses shown by the plus or minus. And these were evoked only when the stimulus was presented in the center of the receptor field. Now, when they moved the spot, they found robust responses when the spot moved in the preferred direction, but very little responses when the spot moved in the opposite or null direction. And this is pretty amazing because it's the same amount of light, but there's a certain computation that's happening that prevents and lets the cell decide which direction the stimulus is moving in. So they also had the brilliant insight to realize that since the motion in the null direction was suppressing something in the center, it must invoke some sort of a null circuitry, okay? And for the last 40, 50 years, what we've been trying to do is identify that inhibitory circuitry, and we've done that quite successfully and identified the starburst amacrine cell as being the mediator of that null inhibition. So starburst amacrine cells are a mirror symmetric population, and here you can see the on and off starburst amacrine cells. They send their dendrites into the inner plexiform layer, and they co-stratify with DS ganglion cells, the direction-selective ganglion cells. They release GABA and ACH from their distal terminals, and importantly, when you destroy these cells by initially done by the cell-specific genetic ablation, or more recently by chemogenetic silencing by Marla Feller's group, you see that you completely lose direction selectivity in downstream ganglion cells. So these cells play a really important role in mediating, first computing direction and then mediating that to downstream ganglion cells where they can be output to the rest of the brain. Okay, so how do they actually provide null inhibition? This relies on two important factors. Okay, so first of all, we have to, people have found that the starburst amacrine cell or SAC only connects to the ganglion cell from the null side. Okay, so this is the DS ganglion cell, it's got a preferred direction, and stimulus coming in from the null side activates starburst first. Okay, so if you, although the starbursts are present right through the dendritic field of the ganglion cell, if you stimulate starbursts over here, they do not provide GABAergic inhibition. And this was confirmed both functionally using paired recordings and also anatomically using serial block EM by Brigman et al. Now, the second important feature is that the Sac dendrites themselves are direction selective. Okay, and they'd like motion from soma to the dendritic tip. So when the stimulus comes in from the soma to the dendritic tip, it evokes a large GABAergic output, which suppresses the glutamate from the bipolar cell. And conversely, when the stimulus moves in the ganglion cell's preferred direction, it does not activate the dendritic tip. There's very little GABA, and the glutamate signal goes through and evokes robust spiking in the preferred direction. So this is the basic fundamental model that we've kind of reached about 5-10 years ago. Now, the other aspect of this where people put a lot of effort is to realize the bipolar cell output is not directional. And this they did by very elegant GCamp imaging of the terminal or measuring glutamate with iGlu sniffers. And they found that the output of the bipolar cell is not directional, although of late there might be some inkling that a few bipolar cells might be directional. But by and large, let's just say the bipolar cell input is not directional. So it's mainly the GABAergic inhibition that's shaping the directional response. Now, I mentioned these are GABAergic and cholinergic neurons. So the question is, what is acetylcholine doing? And right from the beginning, when they realized this neuron was releasing both, this led to the imagination of a mechanism that would invoke, that would allow the starburst to provide both excitation and inhibition for creating direction selectivity. And this is a picture on the left from David Vaney, who's showing excitation coming from the left, from the preferred side, and inhibition coming from the rest of the receptive field. And he imagined that all dendrites of the starburst would either release ACH or some would release GABA, and that, therefore you could get this mechanism. Now he wasn't quite right in the mechanistic details, and this wasn't true, but the ideas were correct. Okay, and later on, 20 years later, work from Jimmy Zhu's lab made beautiful paired recordings to test this model. And what they found was when you made paired recordings from the starburst on the null side, you could evoke large GABAergic inhibition as well as cholinergic excitation, which you measure with a holding potential of 0 millivolts for inhibition or -60 or -70 for excitation. And there you could get beautiful release of GABA and ACH. Okay, so it kind of doesn't support this model. However, from the preferred side, you don't get any GABAergic connection, but you get a robust excitatory current mediated by acetylcholine. Okay, so there's a differential transmission of ACH and GABA to the ganglion cell. And this was quite exciting. At the time, the issue was with these recordings was Jimmy found that in the natural stimulus, ACH didn't do much. So when they blocked ACH, the response wasn't changed very much. In contrast, what we found in the mouse retina was blocking ACH had a huge effect on the ganglion cell response under a variety of conditions. Okay, so on the left, you see the response to high contrast stimuli. This is almost 200 or 300%. It's getting reduced by 50%. If you go to normal stimuli, which are kind of lower contrast, the response is completely squashed. Okay, it goes to 0. For a video of a natural movie taken from a camera on a mouse's head, you see the response is again very strongly suppressed. Okay, so regardless of whether ACH is directional or not, it's really a driving force for excitation in the DS ganglion cell. So this is also, this indicates that if ACH is providing excitation and inhibition to the ganglion cell, then theoretically it should be able to drive direction selectivity. And this turns out to be true and can be tested using an optogenetic approach. So this experiment done by Santosh again shows a recording from a DS ganglion cell under control conditions, then photoreceptors blocked, and then the light intensity is increased a lot to activate the channel rhodopsin that is expressed only in starburst amacrine cells. And then you can see the same kind of tuning properties. And I guess you can see this best over here. You can see that the control tuning matches that of the channel rhodopsin tuning. Okay, and this is for one cell, and this is for many cells. Okay, so you can see the Starburst network itself has all the elements to create direction selectivity. Okay, so to summarize then, we kind of modified the basic model in two distinct ways. First of all, we suggest that it's not the bipolar cell that drives excitation, but the preferred Starburst amacrine cells. Okay, they are the main source of excitation. And now, you might wonder what happens to the bipolar cell input. After all, if you look anatomically, it's the same set of bipolar cells that drive the Starburst and DS ganglion cells. So if they're driving the Starburst to release acetylcholine, why are they not driving the DS ganglion cells? And it turns out they are releasing glutamate onto ganglion cells, but this glutamate is mediated by NMDA receptors. Okay, and this is a kind of neat system where NMDA acts on the ACH and GABA, and it acts in a multiplicative fashion. And this is work done from Jeff Diamond and even previously. They found that when you block NMDA receptors, you don't change the tuning of the ganglion cell. It scales down by a similar factor. So it would multiply a null response by 2. It would multiply the preferred response by 2. Okay, so this is a multiplicative aspect of NMDA. So in this sense, the ACH is and the GABA, the primary drivers, and the NMDA is the neuromodulator. So another last aspect about this circuit is it would allow to generate certain EI offsets between ACH and GABA, okay, which this circuit does not do as well. So, and that's kind of schematized over here. So when you come in the null direction, you activate the null Starburst that release acetylcholine and GABA. So that happens at the same time, basically. Okay, so you have in GABA and ACH coming in by these null Starbursts, and then a smaller delayed excitation from the bipolar cells. If you come in the preferred direction, what you see is this excitation is drawn out to the left, and it's become really offset from the inhibition. Okay, and this is very well tuned as well. So in this model, you don't need the direction selectivity of the Starburst dendrites. In a nice set of experiments done by Laura Hansen, she found a way to manipulate the DS in the dendrites of the Starburst amacrine cells such that they produce non-directional output of GABA. And under these conditions, she could still generate a robust direction selectivity in the ganglion cells. Okay, so you can see there's multiple mechanisms to generate DS in the ganglion cells at a macroscopic scale. Okay. Okay, so all this would be well and good, and we'd probably stop here if we it wasn't for these beautiful recordings from Ben Sevier. So if the excitation and the inhibition were summed at the soma, then our work would be done. But early work from Christoph Koch, just based on the passive biophysical properties of the dendrites and the natural branching patterns, he suggested that the inhibition and excitation will interact nonlinearly in these dendritic subunits. Okay, more recently, Ben Sevier managed to get an electrode in there and measure activity in the on dendrites of a rabbit on DS ganglion cell. Okay, and he found very robust dendritic spikes. Okay, and that's kind of shown over here. So as the stimulus moves in, you see these dendritic spikes measured in red precede the somatic action potential. It means activity was initiated right over here. And as the stimulus moves along, you see the latency changes. And when the stimulus is back here, then it's the somatic action potential that's causing this dendritic action potential to back propagate. What this means is it's pretty exciting because it says that even when you have a computation at a distal site, that information can be reliably transferred to the soma. Okay, in a passive model, the inputs closer to the soma would win out. But if you put a little action potential over here, you can very efficiently transfer information to the soma. The downside of this is if you have misinformation, so if inhibition and excitation are not balanced correctly over here, and the response is not directional, then you degrade the output of the cell. Okay, so having dendritic nonlinearities like this requires the inhibition and excitation to be really tightly coupled, and you have to have this kind of configuration right through the dendritic field. Okay, that's the prediction. Now, measuring this is quite difficult, but you kind of get a sense of how well inhibition might be balanced through the dendritic field, and just by looking at the anatomy. Okay, so this is a diagram from Kevin Brigman's paper, and it shows the receptor field of a ganglion cell in gray. And what these little vectors are synapses, and which point to the soma of the starburst. Okay, so here's the preferred side, and you can see all the vectors are pointing towards the null side. And if you do the quantification of this, you see the vectors of the starburst dendrites are oriented in the opposite direction of the ganglion cell's physiological response. Okay, and this is consistent with the model I showed you. So when the stimulus comes in, this way you would activate inhibition and suppress the ganglion cell. So we have inhibition right through the dendritic field. Although if you look a little closer, you might see there's a systematic shift in the tuning, but let's not worry about that right now. But the important thing is you have inhibition distributed through the dendritic tree. For excitation, especially acetylcholine excitation, the issue is a little more tricky. Okay, and this is because we think acetylcholine might be mediated through volume transmission. So in the retina, the starburst American cells are the main source of acetylcholine. And I showed you this, they're bistratified, and the receptors are all through the inner retina. So that means the ACH has to travel great distances to reach those receptors. Now for the DS ganglion cells, of course, they co-stratify, so the distance doesn't have to be that great. However, if you look at the differences between the functional wiring and the physiology, you kind of envision that the acetylcholine is not synapse specific. So once again, Kevin, these little circles depict the wraparound synapses Kevin found. And you see they're more on the null side, very few on the preferred side. Yet when you measure excitation, you have an almost equal amount of excitation from preferred and null sacs. And if there are no synapses, one imagines that this connection would be through volume transmission, and therefore doesn't need the wraparound connection. Now, if you imagine volume transmission and you realize that there's lots of varicosities within this dendritic plexus, so the starburst coverage is huge. At each point, there are almost anywhere from 10 to 30 dendrites, okay, at each point in this honeycomb. So you imagine that to be a lot of starburst varicosities, each coding a different direction, and this is shown in different colors. And now if ACH spread a long distance, then you imagine the signal at the single dendrite would integrate from multiple varicosities, and therefore the signal at the dendrite should be non-directional and relatively slow. However, if somehow the synapses were more synaptic-like, and there were some synapses that we missed over here, you'd get much more transient input to each point in the dendrite. And since there wouldn't be the spread, the signal at the dendrite would be directional, just because the starburst dendrite is releasing in a directional manner. Okay, so the direction selectivity of the dendritic signal largely depends on the spatial dynamics of ACH, and this is what we sought to determine. Now, the first clue that it wasn't paracrine, or at least the whole mechanism wasn't paracrine, came from a simple recording of the spontaneous activity in voltage clamped DS ganglion cells. And over here, you see just holding the cell at -60 in a cocktail of glutamate and GABA antagonists, you can still see very fast events. And these are quantal mini-like events. Okay, they have a fast rise time and a fast decay time and a very characteristic shape. Okay, their peaks are slightly larger than the AMPA receptor ones we measure, and smaller than the GABAergic ones. But importantly, they rise exactly at the same time as GABAergic minis. Okay, so they seem as fast as GABAergic minis. They might be a bit slower than the AMPA minis. Now, if you put on a alpha-6 receptor antagonist, you see all of them are gone, suggesting that they're mediated by this alpha-6 subtype of nicotinic acetylcholine receptors. Okay, so this is telling us, okay, it can't be all paracrine. There must be some synaptic component to it. So the most parsimonious explanation for those is, okay, of course, null starburst make bona fide wraparound connections. That must be the source of the minis we see. So we said, okay, let's go and test that by making paired recordings. Okay, then the null input should be much faster. However, when we made paired recordings, when stimulating the starburst, we found the response in ganglion cells was quite slow. Okay, so here, pay attention on the rise time. You see this is in black is the rise time of the minis, but the rise time of the evoked are much slower. And this is true even when we drop the voltage that we use to stimulate the starburst and evoked very small responses in the ganglion cell. The responses still remain slow, presumably because when we stimulate the starburst, we have vesicle release all through the arbors in an asynchronous manner. So this made it very difficult to ascertain the properties of single starburst connections. However, at that point, a paper from Marla Feller's lab caught our attention, where they actually replaced the calcium in the bath with strontium, and which led to a desynchronization of GABAergic vesicles. Okay, and this is a trick actually used by Wade Regier and Chuck Stevens to study delayed release at cerebellar synapses as well. Okay, so it's a standard method to kind of desynchronize release from a neuron. So when you replace calcium with strontium and you give a brief depolarization to the starburst, what you see is a brief response followed by this barrage of asynchronous activity. Okay, so riding on this plateau are these really, really fast events. Again, this is done from a preferred starburst that doesn't make many wraparound synapses with the DS ganglion cell. So if you compare the response to the spontaneous activity, you see the asynchronous responses are equivalent to those evoked, to those observed spontaneously. They have exactly the same waveform. Okay, they have exactly the same amplitude distributions and exactly the same rise times. Okay, so it's saying that the evoked asynchronous EPSCs are likely mediated by a single vesicle. You can see also the important fact that when you make these measurements from a preferred starburst, and you know it's preferred because it has acetylcholine and not GABA, you can see again it has the same distribution of peak amplitudes and rise time compared to the null starburst amacrine cells, which release both ACH and GABA. Okay, and you can't see the green because it's just hidden over there. Or maybe in the slide of, sorry, the nulls are in blue versus red, and you can see they're completely overlaid. So this is pretty surprising because you have a situation where you have wraparounds and non-wraparounds, and they're still giving you ACH with the same connections, with the same kinetics, okay, and the same properties. So this was really surprising. Okay, if you're having really fast transmission from preferred sacs, we wondered then, are there synapses that we missed out on this side? When Kevin originally did these studies, he used data sets in which the ultrastructure was missing. These were the first data sets used, and to allow for a rapid tracing of the neurons, he used fixation methods where you couldn't see vesicles and you could just see these clear cylinders, which allowed for a rapid tracing. And he recognized the GABAergic synapses by their wraparound morphologies. Okay, so we wondered if there could be a second synapse on the periphery of these wraparounds, which weren't observed in this data set. Okay, so ectopic, this is called ectopic release, when you have release away from the presynaptic zone. And this has been hypothesized to mediate fast cholinergic transmission in the autonomic nervous system, and also in retinal bipolar cells, and maybe other parts of the brain as well. So ectopic release is an important mechanism, and we wondered if it had any role over here. However, in this, if you go to ectopic release, you imagine that the release would be independent of the main presynaptic release at the main presynaptic specialization. So to test this hypothesis, Santosh measured activity from neighboring pairs of DS ganglion cells. So over here, you're looking at the pair of DS ganglion cells. You see the somas are quite close and the dendritic fields are quite overlapping. And then he asked, are these, and in this case, you can see the they're coding opposite directions. And what you see over here, the minis, and they're again measured in a cocktail blocking GABA receptors and glutamate receptors. And you see every once in a while a mini that's quite synchronous. Okay, so this is kind of really weird, right? I mean, normally a mini is a kind of hallmark of a synaptic localized response. And here we're looking at minis that are synchronized across two DS ganglion cells. If you kind of zoom into that, these synchronized events, you can see them at a higher time resolution, and you can see their rise is perfect together, and they really go, at the same time. So this gave us the indication that a single vesicle releases acetylcholine, but that acetylcholine can spread to two DS ganglion cells and activate nicotinic receptors rapidly. Okay, within a millisecond, you've got your response. So this is really neat, and it's kind of a new form of transmission, so to speak, and we kind of refer to it as multi-directed transmission, because the ACH is being directed to at least two dendrites. It's possible it's even three, but as right now we can only measure from two DS ganglion cells. Now, if you look at the amplitude and decay kinetics, these are quite correlated, which is another surprising aspect. So usually the decay kinetics of a mini is shaped by the deactivation of the receptor. So when receptors bind transmitters, the transmitter, it holds on to the transmitter for a little while, and then the unbinding of the transmitter or the deactivation of the receptors is what determines the decay of miniature events. In this case, you see that there's a very strong positive correlation in the decay kinetics. So if you have a slow event in one cell, you have a slow event on the other. You have a fast event on the other cell, you have a fast event in the other. So, okay, so there's a very strong correlation in the decay kinetics, suggesting maybe they were shaped by a common factor. Now, what this common factor is, we don't know, but maybe it's the concentration of ACH in the cleft or in that local vicinity. That's the hypothesis. Okay, so the next question we addressed is, how far does ACH spread? Okay, so to address this question, we looked at the relationship between the physiological and the dendritic overlap. Okay, and this is a method designed initially by Trong and Ricci. And in this method, what you do is you measure the physiological correlations and then look at the dendritic overlap. Imagine if there's a huge, a perfect dendritic overlap, then all the events would be correlated. And of course, if you have only a little overlap, then you have a few chances for overlapped events. So we computed the distribution of nearest neighbors, and that's what's kind of you're seeing in this figure. So in the hot regions, there's a very strong overlap. Okay, and then this is the cumulative distribution function for those nearest neighbors. And you can see this ramps up very quickly. Okay, but 10 microns is a very high chance that you'd have two dendrites within that 10 micron vicinity. So if AC8 spread 10 microns, then you would expect 75 or 80% of the events to be correlated. So then what we did was we plotted the strength of the correlation against the fraction of dendritic overlap in these different bins. Okay, so if you looked at the first bin and you said, look, you need to have a perfect overlap, then you would see this relationship. Okay, and you see it's underestimating the amount of correlation. However, at 1 micron, when you can consider this bin over here, then you see a good mapping of the functional correlation. Okay, if you go to larger distances, then again, they're not matching. So it seems from this analysis that AC8 spreads about 1 micron from the release site. Okay, so dendrites need to be within kind of 1 micron region, and then you have correlated activity. So this is pretty exciting because it said, look, you've got a condition where you're getting synaptic and paracrine forms of transmission at the same time. And it kind of requires a new definition for cholinergic transmission, basically. So we went to David Burson and told him our results, and he was quite excited. And he dug deep into his data sets and examined these varicosities again with a lot of detail. So he remapped, he contacted Kevin, and he got a new data set, which is published by Ding et al. in Nature, where they had ultrastructural details. And he, again, did the analysis where he mapped out the wraparounds and followed them to different kinds of DS ganglion cells. And basically what he found was when he looked at the varicosity in detail, and in green you see it's making a wraparound synapse that kind of where the GABA transmission happens. Quite often you had a second dendrite in the peripheral region. Okay, there was no synaptic specialization over here. Okay, and over here you might see some vesicles, but normally there weren't even that many vesicles over here. Okay, so there was no sign of ectopic release per se. But quite often you had, at least 50% had of the varicosities had a second dendrite from a DS ganglion cell coding an opposite or another direction passing by within one micron. You looked at the angle of that second DS ganglion cell, you see it tends not to be of the same one that's making the wraparound synapse, okay, but it could be any of the other three, okay? So you have this kind of novel tripartite complex where you have a single cholinergic axon terminal that's kind of has two postsynaptic sites in a very small region. Okay, just to summarize this part of the talk, you have a novel form of multi-directed transmission where a single vesicle release from the Starburst terminal, it doesn't create minis, so it's not paracrine, okay? It's not spillover because spillover means it would go and create a large response in the null and then into a preferred, but you see nothing of the sort. You see the responses in both, so it's not the typical spillover transmission that we refer to, and it's not ectopic release because we see again both simulsynchronous responses in two ganglion cells simultaneously. Okay, so it's similar to paracrine in a sense because the transmitter is spreading a large distance, but the main difference is in paracrine you don't normally have fast quantal responses. Here you have fast quantal responses. And to understand why, we kind of probed a little further into this, and I won't go into the details of this, but I'll just give you the highlight from Jeff's modeling work, where he took, he reproduced the concentrations of ACH that might be experienced in the synaptic cleft using simulations from Barber and Hauser, where you see when you have release at the site, you have a very large concentration of ACH, but that comes crashing down within a few microseconds. Okay, within a millisecond, the concentrations have equilibrated between a proximal and a distal site, maybe 1 micron away. Now, if you simulate this and you ask how receptors would behave, and to do that, you kind of use these reaction mechanisms with rate constants. And again, all this is from a published literature, and we just kind of, for an initial step, we took the values from this paper modeling nicotinic responses in the autonomic nervous system. And what we found is, if we looked at alpha-3 receptors, then they behave very differently at the proximal and distal site. So this could not mediate multi-directed transmission. However, the alpha-7 receptor, and we modify the desensitization kinetics because really we have alpha-6, but we don't have these parameters available for alpha-6. We slow down the desensitization of alpha-7, and what you see then is the proximal and distal responses are much more similar. They both are rising within a millisecond, and they have comparable. This one is a bit smaller, but at least they're comparable. And in black, you see the waveform of the miniature spontaneous EPSC that we measure. Okay, so again, this might be wrong in the precise, detailed parameters, and we kind of trying to optimize that. But the main point is this fast paracrine or multi-directed transmission really depends on the biophysical properties of the receptor. Okay, finally, we're trying to understand what all this means. Okay, why are we going so much and trying to understand why ACH needs to be fast and so local? And to directly get an idea of what the light evoked responses would look like, we then collaborated with Keizuke Yonohara and Akiro, and also Yulong in China, who developed these ACH sensors. Okay, and here Santosh made a recording from a Starburst amacrine cell, and he's expressed the sensor in the whole network of Starbursts. Okay, but he's loaded only one Starburst with a red indicator. When he delivers a pulse, you see a very rapid ACH signal, and you see that those signals all over the dendritic tree, except they always co-localize or are very close to the dendritic varicosities that you see in red. Okay, so here you're looking at these red little spots are the varicosities of a single starburst, and in each one you can see these really fast transients. Okay, and these are showing that starbursts release acetylcholine from varicosities. You don't get release at every varicosity. I would say about 50%. These are early recordings, but at least half the varicosities we can detect sizable cholinergic responses. Now, the average, these are very localized, and if you make an average of these, this is the plot you get, and this has a half width at full max of about 1.5 microns. So, so when this is again saying that the acetylcholine released from starburst is really spatially compartmentalized. Importantly, Akiro then did express the sensor in DS ganglion cells using a certain Cree line, initially called Cree and then OTX Cree, which labels only ganglion cells. And what he saw was something quite striking. So again, all these colors represent the spatial compartmentalization of the cholinergic signal. Now, in this case, when you come with light and stimulate, you get a response throughout the dendritic tree. But if you go and do this over multiple trials, you'll see the variance of the response is not correlated across small sites. So here in black, you're looking at how the correlation of the noise falls with distance, And again, you see that neighboring distances do not share the same variance. They don't share the same noise. Okay, so they're independent units. Okay, and that number that he gets matches up with this spread that we see over here. So what this is saying, in a sense, is even when you come with light, that the responses are quite compartmentalized in these small sections of dendrite. Now, importantly, if you look at the response to multiple directions, and that's shown in ROI 1 and 2, you can see the response is beautifully directionally tuned. Okay, so here, and neighboring sites can have different tuning. This one, the optimal response is over here, and for this ROI, it's over here, and this is actually its null response. Okay, and if you quantify this and ask what is the directional tuning of all these responses, you see it's basically around the clock. Okay, and this is actually expected given that you can go anywhere around the DS ganglion cell and evoke a cholinergic response. But it's saying the cholinergic response is localized and it's not being averaged over multiple varicosities across around the dendrite, okay, at least as reported by the sensor. It's also important to note that the sensor has a an affinity that's similar to acetylcholine receptors, about 2-3 micromolar. Now, this suggested the cholinergic inputs were coming, they're tuned in all directions, okay? So there's no bias towards the preferred direction. So it's saying, again, locally it's providing a tuned response, but globally, really, it's not. So if you imagine the dendrites integrated information from large sections of their dendritic tree, then having these little local tuned inputs would not matter at all. However, just before I get into that, I just wanted to show you that when you block acetylcholine esterase, you lose this tuning over here, suggesting it's really the acetylcholine esterase is what's keeping the response very localized. Okay. So sorry, coming back to the integration, we predict that if it integrates over a large area, you get a non-directional response, but if it integrates over a small area, we get a directional effect. And previous work using calcium imaging actually suggested that the response was being integrated over a small section of the dendrite. Okay, so here, what we've done is these representations of the directional tuning showing these different colors, suggesting that they're independent units. Okay, so over here, this is this section of dendrite, and you can see if you look at the calcium response, again, I guess the side 3 and 4, you can see the tuning is very different. So this one has a response over here, this one's gone, here the peak is kind of going to 30 degrees, and here it's going up to 90 degrees. Sorry, 120 degrees. So they have different tunings saying that they're independent. The signal seems to be integrated over small areas. Now, I must say that these responses are not physiological in the sense they're measured in TTX because of the dendritic spiking. If you didn't have TTX, you would have a dendritic spike and you would get a calcium response through the dendritic tree. And you wouldn't see the impact of local inputs. What TTX allows us to do is to monitor the local inputs, okay? Now, we can argue about whether TTX, the sodium spike, would allow activity to spread. We don't think so, but it probably does to a small extent. Okay, so you're probably not convinced by this that activity is really local, and we did a few more steps to show that what we're looking at, these different colors, really mean local inputs. So in this Herculean experiment, Varsha recorded from a DS ganglion cell, recorded its preferred and null responses, and then in a mouse line where the starburst somas were labeled, okay, and these little dots, I don't know if you see them on your screen, but you can actually see GFP in the nucleus of the starburst amacrine cells. And then she went with an electrode and electroporated them and basically destroyed a handful of starburst shown by these white arrowheads. And she destroyed the ones on the null side that provide inhibition. And when she did that, you can see an increase in null spiking. Once she did that, she then went and imaged the dendritic tree. And most of the tree was still normal because there's a lot of starbursts over here, and destroying a handful doesn't really destroy inhibition. And you can see in red, most of the dendrite is intact. However, a few dendrites lose their direction selectivity. And if you look at this dendrite in particular, you can see on the same dendrite, one response is strongly tuned, shown in red, and the other one is in blue, showing it's weakly tuned. So the inhibition and excitation are very experienced by these two sections of the same dendrite can be very different, even though they're barely 10 microns away from each other. This is just another example of a before and after. So before the ablation, you have a very strong DS tuning around 0.5 in many parts of the dendritic tree. And then when you ablate, in this case, we've lost a DS in most of the arbor of this small section that we've zoomed into, except we seem to have very strong DS at one point. Again, this is an average of 4 or 5 trials, so it's not just some sort of noise, but it's saying that it almost seems like there was one input from Starburst remaining over here, and that was able to create direction selectivity within this 5 micron pocket of the dendrite. Okay, so inhibition is very, very local. This is the final slide in case I'm probably running over time. But over here, what Santosh did was he did the converse experiment where he loaded Starburst amacrine cells, patched onto Starburst amacrine cells and stimulated them and loaded the calcium dye into a ganglion cell. And that's kind of shown in green over here. When you stimulate the Starburst, what you see is very local responses. So again, it's saying, ACH can only spread a little bit. So if your varicosity is far away, you don't see anything. And also that the response is really compartmentalized, okay? So now I must emphasize these are calcium recordings. So you shouldn't get confused with calcium and voltage. Okay, the voltage spread is bound to be much more and not so localized. What calcium gives you is just the peak of the voltage, so to speak. Okay, so it really thresholds the signal, and all you can see are the peaks of the voltage. But I would argue that this is what determines whether a dendritic spike occurs or not, and these are the deciding points where information is kind of nonlinearly transformed. So this, in fact, is the important part, and the calcium, the thresholding by the calcium allows you to view these important points along the dendritic tree. Okay, to summarize, we have conventional mechanisms for generating DS, where you have non-directional excitation and a directional inhibition that's summed at the soma to give you your somatic spiking response. However, what I've shown you just now is that the important steps of dendritic integration, synaptic integration, happen in the dendrites, in these small regions. Okay, our first thought was that this design must be repeated right through the dendritic tree. So you have this directional inhibition right through the dendritic tree, and you have non-directional excitation right through the dendritic tree. This was our initial inkling. However, what we found was the ACH was tuned as well. Okay, so it wasn't the simple pattern repeated through, but different patterns of inhibition and excitation appeared to be occurring in different parts of the dendritic tree. So in some parts, in some sites, you could have purely tuned excitation, and other sites you could have inhibition and excitation canceling each other out as previously thought. Okay, these are distinct mechanisms, right? This is an and not, you need inhibition and excitation. This is an and computation where you just need the excitatory mechanism. Now, there's a huge advantage to this because if half your tuning is already accomplished by excitation, the amount of inhibition you need is now much small. You don't need inhibition at each point. And because ACH is rapid, the path that you need, where you need most inhibition in the null direction, okay, this is going to happen naturally. Okay, in the null direction, you're going to get the activation of these null connections or wraparounds, which might co-release acetylcholine and GABA, and then that would happen in a very synchronous, temporally precise manner, and therefore you can cancel them really easily. Okay, if your ACH was spreading around a lot, then it wouldn't be coming from the same varicosity, and you would have orthogonals also being activated and contributing the response, and you could not have this precise timing between inhibition and excitation. Okay, so this provides a very method to synchronize your inhibition and excitation for cancellation. Finally, you ask, why do you need all these subunits? And if you have these small subunits, what we speculate is that you can have a continuous readout of the direction as the stimulus passes through. So if you had a stimulus coming in and have a quick turn, the DS ganglion cell would respond to that. Okay, but this we still again need to verify. And a final point of speculation, looking at these beautiful super resolution images of the inhibitory sites, we kind of speculate where you have lots of clusters of inhibitory synapses, that's where the conventional inhibitory mechanisms would be implemented. But in other parts of the dendrite where you don't have clusters, don't have inhibition, maybe that's where you get these excitatory mechanisms, and tuned excitation play an important role. And these you see in many parts of the cell, you have long regions where you don't have inhibition. But again, this is just speculation. Okay, so finally, I'm sorry if I ran over time. I just want to, again, I've told you who's done the work. This is our beautiful campus in the University of Victoria, and that's our lab over here. And I just want to acknowledge the funding sources that allowed us to do the work. And again, the collaboration of Kezuke and Yulong and David Burson in allowing us to put the story together. So again, thanks for your attention, and I'll be happy to take any questions.
Thank you very much, Gautam, for this fascinating talk. It's really nice to see that you attempted to explicitly figure out what the role of acetylcholine is in the direction selectivity circuit. We have indeed a couple of questions, and while I go through them and ask you this, the people at the audience could still use the chat to inquire about the topic. So the first one is from Brent Young. Are there any post-synaptic densities that can be seen in the EM dataset in the ectopic RGC, or you don't think that ectopic cells form densities?
Yes, so this is it's a good question, and it's in fact a question that's led the whole field into a state of disarray in a sense, because in the CNS, cholinergic synapses do not often have postsynaptic densities. And if you see a varicosity and you see no postsynaptic density, you assume that the cholinergic signals are being transmitted by a volume transmission. Okay, so this is in many parts of the brain. For us, we did not see a postsynaptic specialization either. If you go to the preferred side, the peripheral synapses that we see with the varicosity, we don't see any postsynaptic specialization. And it's only because we have the physiological recordings, we can say that there must be transmission happening over there. Without the physiological recordings, we would think nothing was happening over there. That's kind of why Kevin also probably came to that same conclusion. So it's only a combination of physiology and the SBEM, you can tell that cholinergic events must be happening at these sites without postsynaptic densities. But finally, we are trying to localize the receptors to realize, that the receptor must be there even without the postsynaptic density.
Mm-hmm. The second question is from Marla Feller. Is it known whether acetylcholine and GABA are packaged in different vesicles? Zoo show different calcium dependence of release, indicating different release mechanisms.
Yes, Marla, good morning. I'm glad you woke up. Yes, in rabbit, GABA and acetylcholine seem to depend on different channels, the N type and the P types, and they might be differentially coupled. In mouse, they don't seem to, we haven't really checked the calcium channel dependence, but they don't seem to depend on different biophysical mechanisms. But this I say because what we did was we measured noise correlations in the ganglion cell. Okay, so we measured using the technique by Caffaro and Ricci, we measured EPSCs and IPSCs near simultaneously and looked at the noise fluctuations. And we saw, when we measured both simultaneously, we saw at every trial, if the excitation increased above the mean, so did the inhibition. Okay, so it seemed like at every instance, we had extra excitation, we had extra inhibition, and on instances where we had less excitation, we had less inhibition. So we seemed like we had co-release or co-transmission of acetylcholine and GABA at the same varicosity. Now, whether they come from a different vesicle, definitely not. Okay, they're co-packaged into different vesicles. So if you look at the spontaneous activity, you kind of get a sense of that as well. You don't see any biphasic events, and if the other experiment we did was we measured correlations, and maybe I can show you this. Yeah, okay, thanks for asking this, because yeah, we measured correlations between inhibition and excitation. Okay, so previously I showed you the EE correlations, and I showed you they were correlated. But if you measure EI correlations, you measure excitation at -60, and that's shown at the bottom over here. Okay, and this is between two cells, and you're measuring inhibition in one and excitation in the other. You see they're not correlated. Okay, if it was happening from the same vesicle, then you would expect these to be correlated. Okay. So, and again, the machinery required to package acetylcholine and GABA are different, and so we don't expect them to be packaged in the same vesicle either. So I don't know if I have much more to add to that.
Okay, before I proceed to the next question, because they started really flooding in, I would like to let you know that in case you don't have open your YouTube live transmission channel, that a lot of people are congratulating you and thanking you for the talk. And now back to the discussion and the question section. Mike Manukin asks, first he says beautiful talk, and asks, at the level of the RGC cell, the driving force on the acetylcholine input would be 2 to 4 times larger than the GABAergic input near spike threshold. How does this relate to response and magnitude?
Yes, so the driving force for whether it's acetylcholine or AMPA is always going to be larger near spike threshold, right? I mean, the way inhibition works is not so much by hyperpolarizing the cell, but through a shunting mechanism. Okay, so this nonlinear mechanism, even though it's not creating a voltage and it's near the reversal for inhibition, it really clamps the voltage at the inhibitory reversal without producing a hyperpolarization. So it's just a matter of relative conductance that's important rather than the driving force. And this is, again, clear for, it's not specific for acetylcholine or AMPA, but this is just the way the inhibition would work, and that's why it's so effective. You have a very large inhibitory conductance in the null direction that allows you to clamp and prevent spiking, even though you don't have the same amount of driving force for both excitation and inhibition.
Mm-hmm. There are certain questions also regarding the locality of like the dendritic segment. Anna Vlasic, really amazing story. I'm still a bit confused about how having many different combinations of acetylcholine and GABA interactions at local sites of dendrites, of ganglion cell dendrites, allows for less inhibition being necessary. Are you saying that in some areas tuning is purely being driven by acetylcholine?
Exactly. So sorry, I went over that a little bit quickly, and if you're confused about it, I probably confused everybody. So the easiest way to imagine this is by just comparing the scenario on the left versus the right. And on the left, you have non-directional excitation, which means to create direction selectivity, you would need inhibition at all sites. On the right, you have a heterogeneous distribution of excitation. Okay, so only some of them require attention from GABA, right? The main ones that need to be suppressed are the ones pointing in the null direction. These are key to suppress. Okay, and this is where your inhibition would be directed. These sites do not need inhibition, and yes, it would come from pure excitation, right? So if you just look at the color, it kind of gives you a sense that you need much less inhibition, right? The other aspect of it is, like I mentioned, is the timing of inhibition and excitation that's not shown in these polar plots. Okay, so even if you had this scenario on the left and the E and the I were not well timed, then the poorer the timing, the more the inhibition you need. Okay, if excitation comes before or after inhibition, it'll be very difficult to spike and to suppress, and you need larger and larger inhibition to suppress it if the timing is poor. Okay, what acetylcholine allows, if it's synaptic and temporally precise and occurs from the same varicosities, then the GABA and the acetylcholine can be released from the same varicosity and have this exact same timing. Okay, and the minis have exactly the same rise time, so you'll have a perfect cancellation of the signal. So in that sense, it minimizes the amount of inhibition required if you have perfect timing. Okay, so these are the two important aspects of why we think inhibition, you need less inhibition in this scenario of tuned excitation.
Great. Given that we are running a little bit out of time, I think the next question will be the last one I will ask you to address, and then maybe we can take advantage of the online seminars and transfer this into the Zoom room that we are already sitting in. So for a casual discussion, and whether people want to ask you in person their questions about the topic you just presented. I will choose the question from Sandra Fend because it's something I also wanted to ask you. Hey Gautam, great talk. Do you plan to look at acetylcholine receptor distribution in the direction-selective ganglion cells?
Yeah, absolutely. In fact, it's too difficult for me, so I passed this on to Kezuke, who's the expert, and he's using CRISPR-Cas to put a GFP tag on the acetylcholine alpha 6 receptor, and then use super resolution to actually see that distribution close to the varicosity. It's an important aspect and needs to be confirmed. No, it's a good question.
I agree, yes. Given that a lot depends on the spatiotemporal dynamics, not only of the release, but also of the reception, it's something that is definitely worth looking into. I would like to thank you, Gautam, for this talk today. I would like once again to thank the organizers and all of you actually being in front of your computers at different time zones and making all this happen. And I would like to invite you, as I already said, in the Zoom room that we are sitting. We will post shortly the link for this in the comment section. Thank you very much.
See you.